other body parts too. That to me is much more my intent when I spend time writing books or creating other resources about foot care through exercise. So I started wearing minimal shoes back in 2008, 2009. They were Vibram 5 fingers, the glove-like shoe. So they were very flexible and thin, very minimal as far as materials go. And I was just thinking about them the other day because someone asked me about my first ever long walk, and I thought about it, and I recalled that it was this hike. It was a couple hours outside of Portland, and it wasn't that long ago. I mean, it was the same time that I started wearing minimal shoes, so 13 or 14 years ago. But what I remembered about the walk actually wasn't that it was my first long walk. I hadn't stored in my memory that as my first long walk. I mean, it was 12 miles. It wasn't that much distance to me now, but at that time it was the longest walk that I'd ever done. But the reason this walk stood out for me is because it was the longest walk I'd ever taken wearing minimal shoes. And then I remembered that I had written a blog post because this walk was a killer, not because of the distance, but because of the trail's substrate. This walk was on a slate covered trail. So having a blog is sort of like having a diary, only one that everyone can read and comment in, telling you things like, "You're great, keep on writing," and then signed by someone's actual name, and also things like, "I hate everything about you, and you obviously don't know much, and you're a bad person." Sign someone's anonymous account with a picture of a cat. So here's a fun fact about me. I don't actually have a journal or a diary. What I write, you get to see, and in this case, revisit. So I'm going to pull out what I wrote about that walk over 10 years. My Vacation by Katie. In addition to hanging out with my sister's what felt like 37 kids while on vacation, I also hiked my butt off. I attempted to wear my Vibram 5 fingers or shoeless shoes throughout my entire vacation, something that was going quite smoothly until my most fit friend, Karen, suggested a 12-mile hike up behind the Columbia River. I was totally in. What a great chance to take my 5 fingers into the rock. This wasn't my first time wearing my super light shoe. I had already logged hundreds of shoeless urban miles on the asphalt and concrete of my city sidewalk. And that's why I was completely unprepared for the fact that I had to tap out around mile 8. Starting off on this slate trail, and yes, I said slate, my flexible feet felt awesome. I felt so healthy, so biomechanical, so smug. My feet were mobile while walking, just as they were designed to be, and I could feel every lump of dirt and pebble and ow, like ow, driving into my ow foot with every step. So I toughed it out until I realized that while I had slightly increased the flexibility of my foot over the last year by wearing more minimal shoes, my intrinsic muscles, these are the muscles between the bones of my feet, hadn't really been stretched or strengthened much due to the artificially flat and debris-free surfaces that I had spent most of my time walking on. The mobility of the foot is extremely complex. The foot's 33 joints allow the foot, say 8.6 times 10 to the 36th power number of unique positions. And just in case math isn't the thing, this means that there are more than a zillion trillion movements that your feet can do. So textbooks note and label 6 motions of the ankle, but it's the parts of the feet that give them potential to move into so many different shapes. The foot's ability to deform over the surface it's walking on is a method of data collection of the body. The way the foot deforms to a surface creates a picture of what is underneath the foot. The foot creates an image in the brain as to what's underfoot that helps the rest of the joints move to balance the body's center of mass, position itself well over the surface contours for optimal balance. When the feet are stiff and weak and constant shoes, just partly not being that way, The body's center of mass isn't free to adjust itself in subtle ways. Instead, you get lumbering, lurching, and unbalanced movement. So I know all of these things in an academic kind of way, but what I hadn't really experienced was taking my new foot muscles, new because they had never been over so many rocks for such a long distance on a long off-road hike that challenged the toughness of my feet. My feet were baby feet. These muscles had never been used before and were experiencing fatigue. You wouldn't make a newborn walk a long hike, but that's what I was doing. After 3 miles, I felt like crying with every step. Every step pushed into these tired and sore muscles. My solution was swapping shoes. Thankfully, I had packed another pair. Still flexible, light, and no feel, but I could finish the hike on stiffer bottoms and reduce the amount of movement the terrain was creating in my feet, and I could walk 9 more miles, no sweat. So my ankles and regularly used foot muscles were fine. It was just the little guys in between my foot bones that were so tired. They had never been on a walk before. For the rest of my 10-day trip, I hiked miles in the Olympic Mountains and walked tons all around town. There was a huge difference in the muscles used on urban terrain, flat and hard surfaces, as compared to the natural terrain with rocks and uneven ground. And if you can't tell, if you're in nature, just use comedian Dimitri Martin's definition. Hiking is just walking where you can see. Oh, and P.S. I peed a lot in the woods just because I could. I love, love, love all those purposeful spots. All other, just a note here from 2022. Doesn't this read just like a diary? And this is a lot of love and a lot of too much information. Okay, back to the article. Other notable mentions were my post-walk super open hips and hamstrings, and I didn't even do any stretching. I just let the natural movements be my program and unwind my body under the clear blue sky. And oh, I have zero menstrual cramps. Hello, open pelvis. And here's another side note. Back in 2022, while this is probably also too much information, although it is on my blog, this was the first time I had really deeply moved my feet, as I just said. And yes, 13 years later, the effect on my menstrual cramps, as in they went away and never came back after this walk, has persisted. So I could say more on that, but this episode is all about feet. So 2010 or whatever. There is a very large barefoot movement happening, which is a wonderful thing. However, we have a habit of taking a natural habit and jamming it into our unnatural life. So long distances on cement or asphalt. This takes the thing that we want, like more natural foot movement, while also creating unnatural flow that in many cases can create fractures and overloaded foot bones. So it's pretty smart. You might need more walking in natural environments or a little more padding if you're on hard surfaces all the time. Shoes have been protecting us from our over-rigid environment for some time, and it takes time, years even, to restore function. So exercise your feet. 1. Start by daily stretching. Massage the heels, midfoot, forefoot, and toes. I have a lot of ideas and resources. You can find them on my website, nutritiousmovement.com/our favorite feet, footwear, and walking resources, which I will link to in the show notes of this episode. 2. Do your foot exercise. Not enough to go read them on my website. You actually have to do them. 3. Understand that the position of the foot is maintained by the muscles of the hip. Make sure you optimize your lateral hip, hamstrings, gluteal and adductor or inner thigh muscle strength. Find those fuller range of motion. Tight hips are definitely going to be limiting your foot function. 4. Get a super flexible shoe with minimal or better yet, no heel. 5. Before jumping into non-shoe shoes, feel with your whole body alignment and gait mechanics. Podiatrists are seeing an increase in forefoot fractures when people transition to normal shoes, and a lot of times they're just not mindful of how they're carrying their whole body and feet. We'll learn more about that. 6. If you get non-shoe shoes, be a walker first. If you do choose to run, build up your walking mileage and do your exercises before running in them. 7. Log some miles on a natural surface with elevation changes and rocky obstacles to get even more of your walking parts moving. The urban jungle is not a natural walkable surface. But the good news is we've got all sorts of foot coverings now to fit your specific needs, no matter where you're. Hiking in minimal shoes can yield amazing. For all of you body nerds out there, this is a way to get more of your body and movement. Yeah. All right, friends, thank you for strolling down my pokey memory lane with me. So here's just my final thoughts. Last summer, I took a hiking vacation in Montana with my sister, which was amazing, and we met a new friend, Claudette. Claudette, if you're listening, this episode was for you. Because we were all talking about how to pack to hike in a new place, and I brought up shoes, right? Like, I love shoes, and shoes are the hardest for me to pack when it comes to hiking in a place that I've never been before, because I really prefer to hike, even backpack and even go longer distances in really minimal sandals, even for difficult terrain. But substrate really matters. So I noted to Claudette, and everyone else that we were with, that websites like All Trails could really use substrate descriptions, because without those, I'm never certain which shoes are best. And the story I just read is exactly why I would want not a rating, but a description. Trails are sometimes made with the assumption that everyone will have thick and tall boots, or boots covering your ankle, right? So to be on a slate trail, all of those little pieces poking into my cover, but essentially sort of bare feet, in the sense that I can feel everything coming through the very thin sock-like covering.
How a ketogenic diet and intermittent fasting differ in terms of brain effects, how exercising while intermittent to the body is comparable with stress. A couple of put out when we have new folks to sign up, just head over advice. If you need qualified, this is a long overdue podcast. I've been a huge fan of your research. As a scientist, you have made decades of contributions to our collective understanding of the benefits of biological stress and the supporting evolutionary theory of why almost all organisms actually need stress to thrive. There's a variety of topics that I know you to be an expert on that I'm really excited to talk about today, including your understanding the place of plant phytochemicals in our genetic responses to them from a practical and theoretical perspective, your take on how the decades of caloric restriction in animals has panned out, and our attempts to translate that to those research findings in humans, as well as intermittent fasting as a metabolic switch that has implications for overall health and brain health. So perhaps to kick things off, maybe you could explain why humans need some biological stress, and how maybe modern day society has made that difficult to achieve.
Yeah, during evolution, organisms evolved in environments that were very stressful, even from the simplest of microorganisms, bacteria, where they had to be able to tolerate changes in levels of salinity in the water, exposure to metals that are potentially toxic like iron and selenium, zinc, and they evolved ways not only that they could resist the toxic effects of the exposures, but actually benefit from them. So, for example, in the case of iron and selenium, we know that now that we need iron and selenium for proper health. But high levels of iron and selenium are actually toxic. And so cells evolve mechanisms where they actually incorporate iron into proteins and use the iron adaptively in ways that help them cope with stress. In the case of selenium, for example, several of the antioxidant enzymes, that is, the proteins in our cells that are able to remove free radicals, those proteins themselves, the antioxidant enzymes, have selenium incorporated into them. So that's one example. Then as we move up the evolutionary tree in the multicellular organisms and animals, they evolved in stressful environments. And two of the major stresses are actually food scarcity and predation, competition with other organisms. So individuals that were able to best handle these kinds of stressors, and certainly food scarcity is a stressor, and animals will starve to death if they don't get food. But that stress of the food scarcity is actually a motivating factor, and nervous systems evolve to overcome food scarcity in many different ways. So those are some examples. And in the case of exercise, individuals whose bodies function well in a food deprived state and environments where there's potential for predation are those that survived and passed their genes on. Whatever it is those genes did that helped them perform physically well in a food deprived state as a survival gene.
So we, modern day, fast forward, live in a much different world where we have access to food 24 hours a day, all day, all night. We don't, necessarily need to exercise to get our food either. We can just, get in the car and drive somewhere, or we can have like to our door. So you talked a lot about how the constant access to food and not having these periods of food scarcity where people are not eating may have detrimental consequences on overall health.
Yes. One way to look at that is that when we have food available all the time, when we don't have the need to exercise to get through life, our cells become complacent. They do not maintain their ability to cope with the kinds of stressors that cause disease. Oxidative stress is one key example, inflammation. So a good example is muscle cells and exercise. During the exercise, it's a major stress on the cells in the muscle. There's a big increase in free radical production. Cells are electrically active. The muscle cells contract. There's ion fluxes that have to be dealt with. However, having been exposed to that stress during the exercise, the cells activate gene programs that help them cope with stress and become stronger and more resilient. So, for example, exercise increases antioxidant defenses for muscle cells. It enhances the ability of the muscle cells to clear out damaged proteins, dysfunctional organelles such as mitochondria, the energy producing organelle in the cell. And as well, there are proteins that are initially called heat shock proteins, but their function is to protect other proteins from being damaged. So all of these beneficial mechanisms are stimulated by exercise. So in a person who's sedentary, they have reduced intrinsic antioxidant defenses, they have accumulation of molecular garbage in their cells, accumulation of mitochondria that aren't functioning well, accumulation of abnormal proteins. And this is also true in brain cells. Main thing I study, nerve cells in the brain. There's evidence that's emerging, some from my lab, some from others, that physical exercise, mental exercise, what you and I are doing now around keeping our mind intellectually engaged, we're right now exercising our nerve cells. They're more electrically active. There's more free radicals being produced in our brain cells right now than there would be if we weren't intellectually engaged. But it's not only okay, it's a good thing, because at the same time, the cells are keeping up their antioxidant defenses, bolstering their mitochondrial function. In fact, we discovered that, and this was originally described by exercise physiology, and it makes sense. When you exercise regularly, your muscles get bigger, and in the case of endurance muscles, better endurance. And associated with that, there's an increased number of mitochondria, healthy mitochondria, in each muscle cell. So that makes sense. That the cells then are more able to generate ATP to support their function. We find a similar thing in nerve cells when they're active, and most of this is from animals. So we're extrapolating. But in animals, we can look more directly at the brain in an intrusive way. And we find that running will exercise, what we call environmental enrichment, where you have the animals in cages where they have essentially like playground type environment where they can maintain their mind more active. And under those conditions, exercise, mental exercise, there's an increase in the number of mitochondria and nerve cells, and associated with that, at least in some brain regions, there can even be an increase in the number of synapses between nerve cells or connections between the nerve cells. So that's kind of the general reason it's important to keep stressing in a good way, in an evolutionarily conserved way, that is, by stresses that can normally come transient, short-term, mild, energetic stress. Energetic stress of spending a lot of energy during exercise, or that more kind of subtle energetic stress. I'm sure we're going to talk about what's going on in terms of pathways that are which are elevated during fasting and sustained exercise. You mentioned you talked to Eric Burgess about his work with enzymes called acetylase, and his work showing that ketones have signaling functions and affect gene expression through modulating these enzymes called acetylase. So fasting does the same thing.
Yeah, maybe we can kind of jump into that. So, there's obviously different types of fasting. There's intermittent fasting, eating, or even prolonged fasting. You want to kind of just maybe briefly describe some of those types of fasting?
Sure. The key thing for your viewers to understand is that when they see intermittent fasting is an eating pattern. It's not a diet. Oftentimes, in kind of the lay press and so on, intermittent fasting will be lumped in as a diet, but it's not a diet. It's halfway there. A diet is what you eat and how much you eat. Intermittent fasting is an eating pattern that includes intermittent periods of not eating sufficient to deplete glucose stores in the liver and cause a switch to the use of fat from your fat cells and the ketones produced from those fat cells. So in a person that eats breakfast, lunch, and dinner and doesn't get enough exercise, every time they eat, they're replenishing the glucose stores in the liver, and they may never tap in to the fat stores, and therefore their ketone levels will remain low because the metabolic switch hasn't occurred. It typically takes at least 10 hours to deplete the glucose in the liver. So if a person eats breakfast, lunch, and dinner and then has a snack around 8 or 9 o'clock, they may get up and eat breakfast and have not depleted the energy in their liver and have not switched to using fat. With intermittent fasting, the approaches that have been used in experimental studies, both in animals and humans, are as follows. One is called daily time restricted eating, where the time window that one eats is compressed into, say, 6 to 8 hour time period. So that means the person would be fasting for 16 to 18 hours, which is a sufficient time for this metabolic switch to have occurred. And scientists think, based on a lot of data, that this metabolic switching is important for health benefits of intermittent fasting, but also maybe even of exercise. So daily time restricted eating is one approach that can result in daily metabolic switching, daily elevation in ketones. So for example, if a person skips breakfast, eats all their food between noon and 6 p.m., and if they were to measure their ketones, they'll find that in the morning they'll start to be elevated. If they wake up and go for a run in the morning, they're already in the so they can actually enhance the effect of the fasting in terms of elevation of ketones, and we think in terms of beneficial effects on the brain, cardiovascular system, perhaps even physical performance, which is an area that's being studied now with intermittent fasting.
Push-ups.
There's very strong
20.
evidence that compared
Squats,
to
15.
females that eat plus snatch,
Good morning, 20.
intermittent
15.
fasting is
So,
beneficial for the
arms
heart,
walk.
the brain, glucose
Push-ups, cover
regulation,
squat.
but it's not completely clear yet whether it's beneficial for athletic performance. There's a lot of interest. Okay, so let's get back to intermittent fasting eating. Another intermittent fasting eating pattern is one that's now called 5-2 intermittent. And this is where the individual, 2 days a week, they'll only eat one moderate-sized meal of, say, 600 calories those 2 days. And the other 5 days, they eat normal. So in that case, 5-2 intermittent fasting, the person will have the metabolic switch occurring 2 days a week, but not the other 5. In fact, this 5-2 intermittent fasting, in a sense, kind of triggered the popularization of intermittent fasting. I'll just take a few minutes to kind of give a whole historical perspective. We've done a lot of work in the 1990s, early 2000s, showing that intermittent fasting was beneficial for the brain and animals. We can talk about that in details. And then we'd also published some work on intermittent fasting, reducing resting heart rate and blood pressure, and having anti-inflammatory effects. And then I was approached by a number of clinical investigators. One was Jim Johnson, who worked with asthma patients. And in 2007, we published a small study where we found that these were overweight asthma patients. We put them on a really rigorous regimen where every other day they only eat 400 calories. That's not something that can be maintained as a lifestyle because it's very hard to maintain your body with that. But in these overweight asthma patients, over 2 months, it had profound beneficial effects in improving their symptoms, their airflow in their lungs. My lab, we measured indicators of oxidative stress and inflammation in the blood, which went down not right away, but between 2 and 4 weeks of initiating that every other day metabolic switching regimen. Okay, so we published that study. Then I was approached by Michelle Hartman, who's in England, works with women at risk of breast cancer because of their overweight, and they also have a family history. And she came to my lab. She's seen our work in animals, and there's also some work in animals suggesting that fasting can be beneficial in suppressing cancer growth. So anyway, Michelle and I designed this study where we took these women, 100 women, and we randomly assigned them to either what's now called 5-2 intermittent fasting, or we had a control group where we had them eat dinner. And each meal had 25% fewer calories than they normally take in. And because we've done a calculation that long-term calorie intake would be similar in the two groups. The group eating 600 calories 2 days a week versus the group eating 3 meals every day but reducing their calorie intake. Over 6 months, both groups of women lost about 8% of their initial body weight. And both groups had improvements in glucose regulation, other indicators. But the women on 5-2 intermittent fasting had a greater improvement in insulin sensitivity and lost more belly fat compared to the group that was counting calories every meal. Then what happened is the producer at the BBC, Michael Mosley, picked up that study when he published it in 2010 or 11, and he did a documentary for the BBC which aired in 2013 or 14. He came to my lab and Walter Longo's lab and Krista Verity's. Then so that aired on the BBC, and then people in the UK got interested in fasting. And then all of a sudden, There's all sorts of things showing up on the internet. So it used to be, before the work in the early 2000s, that if you Google intermittent fasting, top hits would actually be scientific. Now if you Google intermittent fasting, it's just like a bunch of random people who have some angle on it. But there's now, the good thing about that is there's now a lot of interest in mainstream medicine. And I wrote an article at the end of 2019, together with a former postdoc of mine, Dr. DeCabo, a review article on intermittent fasting for the New England Journal of Medicine. And the editors invited us to write the article for two reasons. One, there had accumulated sufficient number of human studies of intermittent fasting, particularly in overweight people, to merit coverage of it. But the second reason was that many physicians are being approached by patients asking the physicians about intermittent fasting. And in some cases, maybe many cases, physicians really don't know much about it. They don't know, that there's actually quite a bit of science behind it, both basic and clinical. And from a practical standpoint, they don't know how to prescribe intermittent fasting to a patient and follow up with them, try to help them switch their eating habits. It turns out that in the studies, the ones I mentioned, particularly the ones we did with Michelle Hardy in England, last round, also other clinical people I've talked to, people, if they can get through the first couple of weeks of switching their eating patterns, say 2 weeks to a month, they will no longer be hungry and irritable, maybe can't concentrate well during the time period that they've previously been eating. So, for example, if they decide, okay, I'm going to start skipping breakfast, for the first number of days, even a week or so, they're going to be very hungry, irritable in the morning. And the reason is it takes time for your whole system, everything to adapt to the new eating pattern. And a lot of that has to do with changes in the brain and the neuroendocrine systems that control hunger, the satiety. And we could talk about that. That's a whole another podcast. But one thing we found in pretty much all of our animal studies where we look at the brain, it takes a couple weeks before we see measurable changes in whatever upregulation of antioxidant enzyme levels, increased number of mitochondria in neurons, increased number of synapses, improvements in learning and memory. Okay, I'm going to stop talking now and then let you ask more questions.
Oh, well, this is absolutely. I have so many questions. Brought up so many important points. Well done. Just talking about this sort of adaptation to starting an intermittent fasting sort of regimen and how you adapt to it and how that a lot of that has to also do with some of the neuroendocrine changes, satiety and hunger hormones. That can have a link to the production of ketones and metabolic switching and happening.
Yes, there is a link, and the ketone. And it's very interesting. In hindsight, it goes back to the first study we published with intermittent fasting, which was in rats. This was the one I was at the University in Lexington. What we did is we took rats and we randomly assigned them to either every other day fasting. And the way that's done is one day you go in to the animal stage, you remove their food completely. The next day you go in, you put back food, as much or more that they want to eat. And the next day you go back in, you remove all those food. So they're going 24 hours no food, 24 hours food, 24 hours no food. Don Ingram, who's a colleague of mine when I was at the NIA, had shown that every other day fasting can extend lifespan up to 50% in rats when it's initiated when they're young adults. So we took rats and had them either intermittent fasting or not, and we did a study where we did 1 week of intermittent fasting, 2 weeks, and then 3 months. And then we have these models where we can cause damage to nerve cells and brain regions where nerve cells degenerate in Alzheimer's or Parkinson's or with epileptic seizures. And we found that the most striking effect was in the mild epileptic seizures where we administer what's called an excitotoxin. It's a very interesting story. It's a naturally occurring chemical that's produced in algae. And its levels are accumulate very high levels during red tide, people who are dry. So there was an incident in Canada where these people who had eaten shellfish at a restaurant